Thank you. It's always nice to come to Milan, even if I live in Rome. So thanks for inviting me. I will talk about autosomal dominant osteopetrosis. This is also called Albert-Schomburg disease to honor the very first publication of this disease by Albert Schomburg. He was a German radiologist, the first university professor of radiology in Germany, and co-founder of this journal. I'm not going to read it, of course, in 1905. So EDO is characterized by osteoclast dysfunction, which means that there is a lack of bone resorption and an increase of bone mass, although these bones are fragile, so there is bone fragility due to poor bone remodeling. It is what is called osteoclast-rich osteopetrosis, meaning that the defect is in the osteoclast bone resorption, whereas the osteoclastogenesis is normal or even increased. It is classified in two forms, but this is a mistake because A.1 is not an osteoclast disease. In fact, it is due to mutations in the LRP5 gene, so it needs to be reclassified in high bone mass syndrome. The genuine osteoclast disease is A.2, and in this disease, 70% of patients harbor missense mutations of the CLN7 gene, whereas 30% carry unknown mutant gene. The cells implicated in osteopetrosis are the osteoclasts. You see here them sitting on the bone. They resorb bone through this specific membrane apparatus that is called the Raffer border, and they express a high amount of a specific enzyme that is called tartrate-resistant acid phosphatase. This cartoon shows you the osteoclast that is polarized on the bone, and the bone resorption is an extracellular event, and the gene implicated in A.2 is this one. The product is called the CLC7 protein. It is a chloride proton antiporter that is absolutely important to charge balance the release of protons in the resorption lacuna. Here, and this makes bone resorption correct. The radiological features of osteopetrosis of A.2 actually are quite peculiar because it's not a diffuse osteosclerosis, but is mostly localized in the skull, especially in the skull base, in the vertebrae, and in the pelvis. And the vertebrae are very characteristic because they are called sandwich vertebrae for the thickening of the sub-end planes that you should see here. And also there is what is called bone-in-bone appearance, is clear here. There is a kind of miniature bone inside the bone itself. So nobody knows why this happens, but it's very characteristic of osteopetrosis. There are no systematic epidemiological studies for A.2, so we don't know much about this disease. The prevalence is thought to be 1 every 20,000 born live. Sorry. And the penetrance is incomplete, 66%, so meaning that more than 30% of A.2 are healthy carriers that do not develop the syndrome itself. The symptoms are mostly multiple low-energy fractures. In some of patients, TENS occur lifelong due to poor bone remodeling, anemia for the narrowing of the bony canals, mandibular osteomyelitis for the low white blood cell count that make these patients very prone to get infections, dental problems because the poor bone resorption impairs tooth eruption, pain, and also hypovision and hypoacusis due to cranial nerve compression. The severity range from asymptomatic, I showed you some healthy carrier, to rarely lethal in adulthood. So most of the patients become adults, And it can be seriously debilitating with a very poor quality of life. Sometimes patients are wheelchair bound and need assistance lifelong. And this is one of the cases of very severe osteopetrosis you see here. So there is an important unmet medical need for ADO2, that is a curative therapy. In fact, the treatments so far are only palliative, surgery to stabilize the fractures, antibiotics for the infections, analgesics for the pains. In the past, they treated the treatment with PTH, vitamin D3, and calcium supplementation, but they failed completely. A potential curative treatment could be the hematopoietic stem cell transplantation, but you know that this is a quite risky procedure, so it has been applied only for a very few extreme phenotypes, especially if diagnosed in kids. Currently, there is a clinical trial on Actimune, that is a form of interferon gamma. They are still ongoing with this clinical trial. And the reason why they tried this is because the group of Professor Mike Econs in the United States published this paper on an animal model of ADO2 in which they treated the mice with interferon gamma. However, you should consider that this is a non-specific therapy. There is a lot of controversy on the role of interferon gamma on osteoclasts. Some authors say that it stimulates these cells, others that they inhibit. And then there are a number of important side effects, including suppression of bone marrow function, flu-like symptoms, very frequent, diarrhea, fatigue, reduced ability to fight infections, and in several patients, confusion. And also, you should consider that, at least in mice, there is only a partial rescue of the phenotype in 8 weeks of treatment. So this is, to me, not the best treatment. What we need instead is a specific, targeted, curative, and safe treatment to fully rescue the phenotype. And in our hands, this is the small interfering RNA treatment. What is the rationale for our study? The protein encoded by the CLC7 gene is formed a dimer. And when it's a homodimer, so when the normal subunits are together, the protein is functional. But if one or both subunits are mutated, then the protein is non-functional or malfunctional. So the idea was that if we were able to design a siRNA so specific to recognize the protein only for a mutant nucleotide, we should be able to eliminate the mutant homodimers, leaving only the normal homodimers. And this gene is known to be haplosufficient, and this should lead to a normal phenotype. So this was our rationale. And we focused on this specific mutation in mice. This is the homologue of the G215R mutation in human, which is considered the most frequent ADO2 mutation. As you see, is a missense mutation. First of all, we needed a mouse model, and indeed we obtained this. This is the protein, and this is our mutation that is located here. And you see here that the mouse model is a true ADO2 model. This is the wild type, and this is the heterozygous mice, in which you see an increase of bone mass also measured as a bone volume, and here trabecular number. So the mouse model worked very well. We needed another tool for working in the mice. Primer pairs that were very specific for the mutant mRNA that should not recognize the wild type, otherwise we cannot distinguish between the two. And indeed, you see here, these are wild type, heterozygous, and homozygous mice. The homozygous mice mimic the autosomal recessive osteopetrosis, so they are lethal in 30 days of life. When we use primer pairs that recognize other part of the gene, you see that you can recognize the gene in wild type, heterozygous, and homozygous. But when we use our specific primers, then you see that the wild type is not recognized. And you see the gene also in the heterozygous and the homozygous. And we cut this band and you sequence it, and you see here that indeed there is the mutation. Okay, with these two tools, just to make a very long story short, we were able to investigate the effect of our specific siRNA in E02 osteoclasts that were generated from the mice. So these are ex vivo experiments in which you see that the treatment with our specific siRNA reduced the mutant protein. The control were treated with scrambled siRNA, and in this case you see that there is no detection of the wild-type RNA. And this is a functional assay. This is a bone resorption assay. This is the wild-type level. The E02 osteoclasts resorb about 30% compared to the wild-type. And you see here that the treatment with the siRNA rescued the bone resorption phenotype. Then we went in vivo, and through intraperitoneal injection, we were able to see that there is a very fast increase of circulating RNA in the mice that were injected with our siRNA. And a pilot experiment was done to identify the best posology. And you see here that 4 milligram per body weight of our siRNA was able to reduce the expression of the mutant gene that persistent for 48 hours. So with this, we went through a systematic study to show the effect of the siRNA in various conditions. You see here, young mice treated 2 weeks to improve the phenotype, 4 weeks treatment to rescue the phenotype, adult mice in which the treatment rescued the phenotype in 12 weeks. It was effective in males, in females, in old mice where we only get a proof of principle because the treatment of big mice is very expensive, and we just checked some biomarkers of bone resorption, and it is I will show you also that is effective not only by intraperitoneal injection, but also by subcutaneous injection. Let me go through this experiment just to show you the best condition that we found in our treatment. This is the experimental design. So we treated the mice 3 months old for 12 weeks. After 2 weeks, we took the blood for some evaluation of biomarkers. And so let's start with this. And you see here that the CTX, that are a marker of bone resorption, was decreased in E02 mice and returned to wild-type level upon the treatment with a significant increase of bone resorption. In this blood, we also, from this blood, we isolated the PBMC, and we showed that the treatment was able to decrease the expression in these mononuclear cells from the blood, very clear as is shown here. After 12 weeks, we sacrificed the mice, and you see here, this is the situation in the E02 mice, much more bone compared to the wild type, and this is the situation in the treated mice. So they returned to the normal phenotype. This was also confirmed by the measurement of the bone volume, the trabecular number, and the trabecular separation. We didn't find any change in the trabecular thickness whatsoever between the wild type and the E02 mice. We also rescued the osteoclast variables. This is an osteoclast-rich osteopetrosis. You see that the number of osteoclasts in these bones is much higher than in the wild type, and they returned to normal value with the treatment. This is the osteoclast number, the osteoclast surface, but most importantly, the eroded surface, so the bone resorption, was increased. We also rescued the bone quality. We performed some indentation analysis, and you see here that these two parameters are increased in the E02 mice, indicating a poor low quality, and they returned to normal level with the treatment. Of course, we were concerned about potential side effects, but you see here that the macroscopic aspect of vital organs such as liver and kidney is normal. The weight of these organs is normal. Histopathologically, we didn't see any obvious changes in liver and kidney. Uric acid, urea was not changed, not significantly changed. The GPT liver biomarkers also. We only found a reduction of the pancreatic amylase, but we look in the literature, there is no pathological meaning of this change. And very interestingly, the creatine kinase that is increased in several mice and also in humans returned within the normal level, although the changes is not significant due to the high variability, but in all mice it was returned to normal levels. And we didn't find inflammatory response, which is one of the concerns for the siRNA treatment. Therapy. You see interleukin 6, interleukin beta, TNF-alpha, and interferon gamma. We only found a reduction of TNF-alpha, not an increase. So the therapy is safe on this aspect. It's also specific because not only it doesn't affect the normal gene, but it doesn't affect also closely related genes, such as the CLCN3 and 5. So it seems very specific. Okay, just to give you a translational meaning of this therapy, I want just to show one data about the subcutaneous treatment. Because of course we are not going to give intraperitoneally the drug to the humans, but in the future the subcutaneous treatment could be feasible if you consider the insulin treatment. I think that this is absolutely feasible. And so this is the treatment. These are young mice treated for 4 weeks. 4 weeks in young mice are sufficient to rescue the phenotype. By subcutaneous treatment, you see here a return towards the baseline. And we have all the other data, but I'm not going to show you. So together with the potential evaluation of the efficacy by blood tests that I showed you before, the results by subcutaneous injection makes the therapy theoretically suitable also for clinical trials. There is an important aspect in E05.2. It is not really only a bone disease. There are no epidemiological studies. We really need them. We have a very small cohort of patients, and you see here that a few of them have cognitive alteration, several hematological alteration, kidney alteration, spleen alteration, and muscle alterations. So we looked in our animal model to phenotype other than the bone phenotype. In fact, the CLC7 gene is expressed in many organs, as is shown here. And what we observed very carefully upon the trichrome staining is that in these organs, you see here lung, kidney, and muscle, there is a perivascular fibrosis that is typical in our mouse model. Also in muscle, in which the expression of the gene is very low, as shown here. This is, in some organs, a primary defect. In fact, in lung, for instance, the protein is expressed in the bronchial epithelium and in the alveolar macrophages. And if you looked at the homozygous mice in which the disease is lethal, you see here that there is a severe atelectasis, the alveoli are completely collapsed, and the closure of the airways. So meaning that the mice can die also for these important changes, not only for the bone disease. In these organs, we also found an increase of macrophages. These are F480 marker that is typical of macrophages. You see here in lung, here in kidney, and you see in muscle that the macrophages appear in the muscle. I remind you that the muscle cells do not express the CLC7, but the macrophages do. And in fact, in these mice, in these organs, we found an enrichment in the macrophage signature and an enrichment in the fibrosis signature. And this fibrosis signature is mostly due to the TGF-beta pathway. You see here that the collagen is increased in all these organs, and also at least one of the three isoforms of TGF-beta are increased, and the downstream signaling associated with the fibrosis are also increased. So this could be the reason for the fibrosis, and the macrophages are known to be implicated in the induction of fibrosis in the organs. And this is another result that confirms this. This is the SMAD1/2, that is the transcription factor activated by the TGF-beta. You see here in lung that there is a phosphorylation of this factor and the nuclear translocation. So it seems that the TGF-beta is involved in the perivascular fibrosis that we observe in these organs. Now, going back to our therapy, very interestingly, the perivascular fibrosis found in lung, kidney, and muscle is rescued by the treatment. And this is the measurements. So it is effective not only on bone, but also on the other organs. This is important because a concern that I heard is about the targeting to the bone, but we don't need a targeted therapy. We need a systemic therapy in ADO2. Also, we observed some cellular defect in the cells taken from the mice. And just again, to make a very long story short, you see here that the colocalization of the protein with the marker of the cis-Golgi and the marker of trans-Golgi increases, suggesting that this protein stuck in the Golgi. So the transition from the Golgi to the other organelles is impaired. And this is demonstrated by the fact that, in contrary, the colocalization with LAMP1, that is a marker of the downstream lysosomes, is reduced. And the acidification ability of the lysosomes is also reduced. And also, here, you see here that the intracellular vesicle acidification is impaired in these cells. These are monocytes, but we observe this also in osteoclasts and other cell types. Another changes is an increase of the autophagosome marker LC3B. The expression is increased. These are isolated cells, but in these cells, the treatment, in vitro treatment with the siRNA, in this case, I show just the autophagosomes, is effective. In fact, you see here that there is the reduction of this autophagosome marker. We observed similar changes in vivo, in the lung. You see this marker is increased in the kidney and in the muscle. We also observed the other changes that I showed, but for a matter of time, I'm not showing all the results. And the treatment also is effective here in lung to reduce these cellular changes. So it is really effective at every level. So since we want to go to the patients, we protected the intellectual property. We have a patent in this. And this is where we are and where we want to go. So we made our discovery in basic research, and the preclinical studies, still ongoing, but quite advanced. What we need now is the preclinical development up to the phase 1 and 2 clinical trials. Of course, the level of funding here is too high. We cannot do it by ourselves, so we need a partnership with the industry, for instance, with the biotech. And then, if we are able to go to good results for clinical trial 1, phase 1 and 2, then we can go to the commercialization and post-marketing surveillance, but we need really large capitals, and we are negotiating with the industry to get there. Why this? Because we want to give patients and their families a hope for their future. And I want to acknowledge my research group. This is not Sweden, this is L'Aquila, center of Italy, 1 hour drive, 1 hour, 20 minutes drive from Rome. Here there is the highest mountain in the Apennines, almost 3,000 meters. And I have to thank especially Antonio Maurizio, Anne Curley, and Mattia Capulli for performing this study. And of course, I'm very grateful to our funding agencies. Thank you.
Thank you.